We are examining a fresh crop of Termitomyces fruit bodies from Goa University campus here on 10 July 2015 in Mycology Laboratory of Department of Botany, Goa University, located in Talegaon campus in Tiswadi Taluka in North Goa district of state of Goa in India. This is a plateau ecosystem where laterite is the main rock, and here we find lot of these mushrooms during the month of July and August. Now that has given us certain advantage as the samples are just outside our laboratory, so we can pick them fresh. We can study their ecological and phenological characteristics, which is very difficult otherwise because people have to go to forested areas for collection of these specimens. But here it is possible to bring them and examine them in lab within a few hours, and taking advantage of this opportunity, we want to record certain important aspects in this video clip regarding some ideas on species plasticity in genus Termitomyces. When I first encountered this specimen in the field among the bushes below a Bombay ceiba or say cotton tree, the entire area was enriched by hundreds of kilograms of leaf litter, which falls from a 10 meter to 12 meter high, around 60 to 80 years old Bombay ceiba tree. So there was continuous output of the litter. Now these termites are mostly subterranean and there wasn't any visible mound. So this fruit body was simply growing on the land, on the ground, and surrounded by various type of herbaceous vegetation. This has broken through relatively soft soil and this has emerged in shade. This was not directly under sunlight. So if you look at the fruit body, we can see that the margin has been completely folded. It has been completely recurved. Now the very meaning of recurving is not only the edge but right from its attachment to the gills. The gills, the full gills, the complete folding is there, and not only folding but incurving is also there. So there is first vertical bending, and then you can see this curve. You can see this curve on the other side, and you can see this curve. So there are 1, 2, 3, 4 different, the semicircular folds. Now the 4 different semicircular folds have created this type of projections 1, projection 2, projection 3 and projection 4, folding now this densely situated lamellae here. They are all very crowded and overlapping and almost sticking to each other. So it gives a idea that what exactly is the meaning of species plasticity in Termitomyces, because next to it was a smaller fruit body like this, coming from the same mound probably, because both of the specimens were close to each other within a few centimeters. So from the same mound, how is it possible that you get a fruit body like this and you get a fruit body like this, and what are the conditions which create this type of morphologies? So the concept of definition of the species is getting more and more clearer in genus Termitomyces, with lot of work in molecular taxonomy, in Liophyllaceae, in order Agaricales. But still the fact remains that when I looked at this specimen in the field, I thought that it could be a new species. The reason was that I have never seen in last 30 years of my field work a mushroom specimen of Termitomyces of any species with this type of vertical folds, because these vertical folds are so clear. You can see from the bottom that the attachment of stipe here means it is almost vertical. So this is not only the going beyond the definition of the recurved or reflexed margin, because not only the margin is here. Margin folding is different than what is known as the entire the hymenophore folding. So it is almost symmetrical folding, which is giving a very peculiar type of morphology to this specimen, creating a sort of a pocket, a hollow pocket here, which is almost rectangular. And here water can accumulate, and it can even create a small type of microhabitat for other species, because if the rainwater accumulates in this, as it normally it does, then other species could also grow inside that rainwater. Because although this is an ephemeral species, this is now 2 days old specimen, which we have preserved under low temperature for further studies. But when we picked it from the field, it was very fresh, but infested with various types of mycophilic, termitophilic beetles. And the beetles have caused some damage. So the concept of species plasticity, I believe, may be driven by certain environmental factors, such as disruption or the discontinuation or a pause in the seasonal monsoon rainfall, the increase or decrease in temperature, probably wind speed and direction, and factors like high or low humidity in the ambient environment. But we really don't know, because it's very difficult to take a time-lapse sequence of the folding. So if a taxonomist looks at this specimen, then the compulsion would be, why not diagnose it and describe it and identify features which are totally different than what are described in the standard taxonomic species key, and then try to identify it as a new species. But we believe that the concept of different taxonomic species in Termitomyces, as has been understood right from Heim to Botha, Eicher, and so many other scientists who have worked on termitophilic agaricales in paleotropics, they are of the opinion that macromorphologically the species may appear different, but micromorphologically there is a lot of congruence, there is a lot of similarity as far as reproductive organs like basidia, basidiospores, and the cystidia and other features bring them very close to each other. But mostly the descriptions are based on macromorphological characteristics. So how do we then define this fruit body, which is folded, which is vertically divided into 4 or 5 pockets in a curvaceous manner, and the entire most important, biologically, genetically most important aspect is how then the species ensure basidiospores dispersal if the gills are folded in an unusual manner, because this is not the way the gills are supposed to be that exposed to rain and wind, because there is possibility that the spores could be washed by rain, and that is not the way to the normal Termitomyces species. They are supposed to be similar to simple umbrella like shape, where the spores are discharged mostly in this zone in a sort of a imaginary circle, and then whatever the worker termites can forage, they could transport these spores inside the nest and sow those spores on the fresh fungus comb to start a new Termitomyces culture, which they maintain in an asexual form. So this much is known, but the difference between this hymenium and this hymenium we can see very clearly. So we cannot compare this as an entire margin, and this is a folded margin. Both are Termitomyces, both have come from same ecological space, same habitat, same fungus comb. So how do we explain? So we cannot call these as two different species. So this seems to be an abnormal feature or a variant of this, because this is generally the normal behavior of Termitomyces with the intact cap, NS type. but never in this manner that we have seen today. So it is a dilemma of sorts when we think that this could be a new species. We really have to collect strong evidence on basis of taxonomic diagnosis of the crowded lamellae, which are pinkish. Then we have to look at the micromorphological features like nature of hymenophoral trama, then subhymenium, whether it is filamentous, well defined, or it is cellular. Then we have to look at the sterile elements like cystidia, their shapes, their size, their distribution, their polymorphy. Then we have to look very carefully at type of basidia and ensure that they are all normal, tetrasporate with long sterigma. And then we have to examine various types of basidiospores to see whether only one shape is produced by this organ, and whether there is a lot of polymorphy among the basidiospores which we can release from processing of this lamellae. We have not been able to obtain a very good spore print because it's difficult to obtain a spore print from this folded specimen. But after analysis of these, perhaps if we find the very strong evidence that what we see here is also seen micromorphologically, then only perhaps it is possible for us to lay a claim that we have encountered from the same mound, from the same fungus comb, these two different species. And Pegler in Kenya had speculated on species plasticity in Termitomyces because same thing he had encountered in Kenya, I mean almost 20-25 years ago. And that paper in I think Zambia, where they had done the work on Zambian Termitomyces species, they also were surprised when they collected specimens that from almost same fungus comb, two different species were emerging. So here we really don't know till the time we process this and collect further information. Right now, for the purpose of recording this, we believe that this form is created by environmental features, and this may be a larger variety of the normal Termitomyces species which we see in this. So if we compare both micromorphology, morphologically nothing new may be found of significance for taxonomic purpose, but macromorphologically, ecologically speaking, we have found a tremendous degree of divergence, tremendous degree of change, tremendous degree of plasticity when we compare both these forms. For example, if you look at the ambonal region of this, now the whole fruit body is hemispherical, it is convex, and then I don't think that it is going to be recurved like this. So the melanized part that we can see here, which is like a thick layer, leathery layer, rough but centrally sticky, is oppressed, means it is flush with the surface of the fruit body. In this case, we see here that a totally different picture emerges here, that it has the melanized part is also present there, but it is flat and it is slightly distorted. So what is the reason? So the reason could be that this is draining the water, whereas this is covered by water. So ecologically speaking, this is the much better design because it doesn't allow stagnation of water. The water discharges from this black ammonal part when heavy rains are there, and it doesn't cause decomposition of the fruit body. Neither it is favorable to the insect because this is the toughest part for the mycophilic insects to drill through these, or the gastropods or those mycophilic snails which at night graze on this. They find it tougher to break through this, so that the specimen is protected in nature. In this case, it is something which is not beneficial to, unless we know the reason, something which is not beneficial to the fruit body. The reason is the water will get accumulated in this part and it will be like a small pool of water, which is quite unusual for Termitomyces species. In fact, we believe that at least we have reported this for the first time, this type of hollow pocket created by folding of the edges of the natural folding of the edges of the root body, so that we have a hollow here where water can get accumulated with heavy rainfall, and then it will cause lot of other issues also. But at the same time, we have also noticed the highly tough nature of the epicutis. Now that is not there in this as much as it is encountered in this. So we wonder whether this is a form which is perhaps created to withstand vagaries of climate due to global warming. This is a big question mark before us. And whether this type of forms will be common in genus Termitomyces, especially in state of Goa, wherever we encountered oddities like this, morphological oddities, because for us it is still an oddity, because otherwise we cannot just explain these are the normal shapes to which we are used of Termitomyces mushrooms. But when we encounter an unusual large fruit body like this, the question is, what is the purpose of this folding, why it is happening, what are the driving factors, and what is the explanation for these driving factors, and how this species, which is genetically programmed to behave like this, suddenly transits into this stage, because this needs immense amount of energy, cellular energy and metabolic energy also, to cause this folding, because it has to lead to specific signaling by the mycelium to create this type of stress. Now this part is completely stressed wherever the curve is there, biomechanically speaking. First of all, the biomechanics of the developmental aspects of mushroom is not fully understood. A lot of studies have been done by Alan Reiner, by David Moore and other mycologists in England, but even now we have very little understanding of the biomechanics of Termitomyces folding. So we can only speculate, and we can assume that a lot of energy is involved. This was not one step process. Over a period of days, I think this process of recurving or folding was taking place, creating this fold, and same way the other folds also. And it is possible that all this folding was taking place simultaneously. It wasn't like this fold took place later and this fold took place first. So it was uniform, recurving or folding. That means a lot of mechanical stress was transmitted for the purpose of uplifting and folding of this reproductive region of the mushroom. Now what science can tell us about these forces, we are totally in dark about it, although it is possible to model this mathematically and understand that all this turgor pressure, or what we can say as the hydrodynamic pressure, is provided by the thick bulbous type. We can see this type here. This type is like a cylindrical organ. It has only the biomechanical purpose of supporting this shape and this biomass and this weight, and it doesn't grow further. After it attains a particular height, this is the optimal height perhaps for the discharge of the basidiospores, because elsewhere also we see that this is the pseudorhiza and this is the stipe, and this is the pseudorhiza part here and this is the stipe. So it is around 15 centimeter, this is around 7 to 8 centimeter and around 2 centimeter to 3 centimeter thick, with universal veil remnant here left as a oppressed ring. So this type must have provided a lot of turgor pressure for this context here tissue to fold, and it has caused cracks. You can see here this is a crack which has separated the gills here. So that means there is a natural damage to the entire fruit body. Here there is no crack, there is a very smooth folding here. Then there is a very smooth folding here, there is very smooth folding here, there is no crack. You can see the margin of the gill is intact, except in this place where it has cracked. So the stress was very heavy in this area. So what it means is, biomechanically speaking, the context doesn't behave in a uniform or isotropic manner. So this issue of isotropism also needs to be thoroughly discussed in mycology, because we don't know much about the mechanical aspects of various types of hyphae which are involved in the architecture of this type of design where we have the context tissue here and we have the reproductive section, that is the gene. So what type of hyphae are capable of inducing such type of change, that is right now beyond our grasp, but further research is required into this phenomena, which may give false positive, like from a distance it will appear to be okay, this is something oddity, so it could be a new species and let us go and diagnose it as a new species. We keep our fingers crossed on this judgment, but for time being it would be suffice to record here that we are encountering here a oddity, a mycological oddity in genus Termitomyces as compared to the wild type common form. This form, which we prefer to call Termitomyces heimii, T. heimii, which was erected by Dr. Natarajan of University of Madras way back in 75, and it was named after Roger Heim, the famous termitomycologist from France, who has given us a wonderful monograph on genus Termitomyces in French, and which we regularly refer. So even he would have been very shocked to see the variation in heimii, and we really don't know what to make of this. So here we conclude this part of understanding what we suppose, what we hypothesize as a novel form of Termitomyces heimii, but we need further details to explain these phenomena and also to come out whether this type of behavior will be common with change in rainfall pattern, change in temperature, and what are the basically epigenetic, epigenomic aspects on how this particular feature is now coming up in more and more such deformed fruit bodies, because this is not a chance encounter, but earlier also where we have not really fully documented these forms, we have seen some interesting variations in fruit body morphologies of Termitomyces in our campus, but we have not done extensive survey all over state of Goa or Western Ghats to collect more odd specimens. So this is a mycological oddity, this is an oddity in Termitomyces, and this oddity is being recorded for the very purpose that it should open some discussion regarding what is the role that climate change could play to induce this deformation, because this deformation is not induced by any other force. It is induced by definitely the external forces and the internal commitment of the mechanical folding. So I believe that in presenting this evidence before the world from India, and especially the state of Goa, where there is a lot of diversity of species from genus Termitomyces, we have got an opportunity to examine similar phenomena in other countries in Asia and Africa where Termitomyces species are found in large abundance, and subject any oddities like this to detailed critical biological, ecological, taxonomic and even molecular analysis to come out with a definite idea of how these oddities are emerging in nature.